Experience working in the pharma industry up in the Chicago area, but remains very true and loyal to the Orange and Blue. So thanks for being here.
Great.
Your lapel. All right.
Thank you. Thanks, Laura. Thanks very much. I appreciate the kind introductions. And as Laura said, Rich Johnson is on the phone, and I'll have Rich speak in just a couple seconds here. But I just wanted, by way of introduction, we're going to be talking about CMC strategies and how to develop a CMC strategy for the product that you're developing. Now, what does CMC mean? It's chemistry, manufacturing, and control, which is a very important element for pharmaceuticals and biopharmaceuticals as you're developing those products. But you'll see that actually the principles for CMC are applicable to any really chemistry or biotech products that might be developed, whether it's for ag applications, food sciences, or any other applications. But the principles themselves are really part of a fundamental development for any pharmaceutical and biopharmaceutical, and is part of the expectation from the FDA. More importantly, though, it should be your expectation to create a quality product through the CMC strategy that you have for a particular product. And also important for your investors who will look to see whether you've done a good job of estimating the costs and what's going to be involved in terms of developing a CMC strategy for your particular product. So that's what we plan to address today. As Laura said, my name is Ed Moore. I'm an EIR with Enterprise Works. I'm also a visiting scientist here at the University of Illinois, as well as president and a consultant for BioPhia Consulting, that's helping all medical products, pharmaceuticals, biopharmaceuticals, and medical devices. So Rich, who's on the phone, Rich, would you like to give a short introduction for yourself, and then I'll switch slides and you can take over.
Okay. So good afternoon, everybody. I apologize for not being there in person, as I said, there were some travel considerations that limited where I could go. But I think the GoTo meeting app we use quite a bit, and I think it should work pretty well. So, and I'll rely on you if people have questions that they want to ask for any of the slides that I'm going over. Just tell me to shut up, and I'll listen to the questions and try and answer them. I think it's good to let people have this as an interactive sort of a session. So my background is protein biochemistry. Got my PhD working on an enzyme, but I did a postdoc in the lab working on a system called complement, which is part of your innate immune system, and it got me into immunology and spent a lot of years doing immunology. I came to Baxter Healthcare and worked with the blood and blood products and the interaction of blood with various medical devices. But I also worked with their stem cell group and the biopharmaceutical division quite a bit all through those years. So that's the kind of background that I've got, largely in the early stage development and discovery areas where I spent most of my commercial career. So with that introduction, Ed, if you want to, do I have control or are you going to move it?
I am going to make you the presenter. Hopefully this works. If not, I'll flip the slides for you. So you now have control. If you want to share your screen, Rich, for the slides. Are you still there, Rich?
I am. Oh, yeah. So
Okay.
I hit the share button.
And now just have to put the slides, click on the slides on the PowerPoint. Where's your PowerPoint? Where's the slide? There you go. Now it just...
Okay. So we're going to go back to your version whenever we get to the transition?
Yep.
Okay.
That way you can control the slides. If you want to make those, put those in presentation mode. You got it. There you go.
All right. Okay.
No, wait. We can't see those here. Why can't we see those here?
So why don't I stop sharing and I'll just say next slide.
That's good. We'll do it that way. Sorry, it's a little technology here. All right, Rich, go ahead. We've got the next slide up.
Okay, thanks, Ed. So, as Ed indicated, if you're developing a small molecule drug or a biopharmaceutical, that's what's really relevant to the CMC strategy that we're going to talk about today. But even if you're developing a medical device, there's a lot of interactions you have with the FDA, and I think some of the things that are discussed today will still be somewhat relevant to you. So, next slide.
Okay.
So CMC is actually part of the process of allowing you to move into clinical development. And to do that, the company or the clinical investigator needs to submit what's called an investigational new drug package to the FDA. And it's got a variety of components which are listed here. So you can see the different elements, but CMC is definitely a major part of that package that goes in. So that's what we're going to focus on today, is that one package. And sometime later, and I also have given talks about the IND process, and so we can cover some of these other topics in subsequent workshops if that's of interest to you guys. The next slide, Ed. So this is a cartoon that we put together for a course we taught a number of years back. We've used it over and over again. It describes the entire process for drug development, whether it's a small molecule or a biopharmaceutical. A lot of these steps are relevant. And one of the points that we'd like to make, you think, when you get to the IND stage and CMC is part of that, and that's when you start putting a package together. But in reality, and we'll make this point a number of times, you're really collecting CMC relevant data all the way from the very beginning of this discovery process. So you have a lead compound that you've identified, it's a small molecule or a biopharmaceutical, and you're already just working with it, developing information about how stable it is and whether you need to formulate it in a certain way or some other aspect of its properties that you'll want to incorporate into that CMC package. So CMC is, number one, something that you're collecting information about from the early stages of drug development. It's not something that just happens after you do those preclinical animal studies and ready for clinical trial. The other aspect of the CMC in general that you have to keep in mind is that it is a living document, so it changes over time. There are certain things the FDA will not require you to have in that initial CMC package when you're starting phase 1, but may be required or will be required at the end of that clinical development process. There are maybe changes that you make to labeling that you'll have to incorporate into a CMC document down the road. And as you get out into the market, and there are things you learn about in the general population that are relevant to the CMC, it can change again. So it's really a living document that starts early in the development process and continues to change even after you're out. Next slide. So this is information that really comes from the IND description on the FDA website. So in your IND application, you want to have information about your drug substance and your drug product. You want the composition, how it's made, it's manufactured, what are the controls that are used to ensure that it has, you've identified a property, you've instilled the relevant quality method, make sure that it is what you say it is, it's pure, and what the strength is. And so those are attributes that the FDA requires you to define for your drug. And since they know that methods of preparation change, the phase 1 submissions generally are focused on the raw materials in the new drug substance. So what's this novel drug that you've got? What are its properties? So, but you're going to have to come up with those final specifications by the end of the clinical trial. So you need to have them. You just may not need to have them in phase 1. Next slide, then. So the CMC section contains a number of things, and we are going to go through all those things probably a little redundantly. I'm going to cover some general list of things, and then we'll start going into a little more detail, and then we'll take over the areas where he has a tremendous amount of experience. So we'll go through some of this stuff in more detail down the road here, but just to give you a general overview of what's generally included in the CMC section. So you start off with an introduction. The FDA requires this. And it wants you to state what you believe that either the chemistry of the drug substance, that investigational new substance, or the product where you take that drug substance, formulate it, and package it, whether either of those in the manufacturing or in its inherent properties provides any, it says, signals of potential human risk. So is there anything that's going to cause a problem when you start doing clinical trials? First, we need to spend. So you need to discuss what those are, how you're going to monitor them, or why they may not be relevant to the particular population. You also identify any chemical or manufacturing differences between the drug product that is going to be used in those human clinical studies and what was used in the animal toxicology studies. Ideally, if you can do it, you can manufacture a large enough batch to be able to get through the toxicology work and preclinical studies you need to do and have that same product used, that same batch used for the first phase 1 studies. That's ideal, but generally that's not real practical. And so you have to demonstrate that there's no differences between what you showed to be safe in the animal studies and what you plan to use in the first human studies. So that's part of the introduction. And then the second part of this is a description of the drug substance. What is its physical, chemical, and biological? So where is it made? Number 3, the name and address of the drug substance manufacturer. Next slide, Ed. The general methods for the drug substance preparation, a brief description of the manufacturing process. And that includes lists of reagents, solvents, and catalysts. So these things may not be in the final product, but they're used in the manufacturing, and they need to be included in the description. They like to see flow diagrams. It's a nice way to summarize things. So what are the acceptance limits and analytical methods used to assure that you've got what the FDA is requiring in terms of identity, strength, quality, and purity of that drug substance, your actual drug? You briefly describe what those test methods are, what are the acceptance limits of those different types of tests. If you're getting this done by a third party, certificates of analysis are great to have. So they're highly suggested. Validation data is not required during some of the initial stages, but will be required as the clinical development process proceeds. So assays need to be validated. You could do it as early as you can, but it's not required for everything at that initial stage. And then stability. So you need to provide some information. The drug substance that was used for those toxicology studies and proposed for the human clinical studies is stable over the course of those studies. When we used to do preclinical animal studies in Baxter, the guys would set aside a portion of drug substance stored as the test substance was being stored, and they would go through some analytical procedures up front, and then after the studies were completed to show that there wasn't any change. That's the kind of information you may need to provide. Next slide, then. So we go from the drug substance, pharmaceutical ingredient, to the drug product. So now you change things by adding excipients, for instance, buffers and things to stabilize it, for instance. And then there's you're putting it in something. You have a container of some sort. And you list all the components that may or may not even appear in the final product. Right? So there could be solvents that were used in the manufacturing, or solvents that were used to close the container, or so forth. The name and address of whoever is manufacturing that final drug product, a brief description of the manufacturing methods and packaging procedures. So flow charts again help very much. And here you need to include the sterilization process. So if it's a biopharmaceutical, is it aseptic fill? If it's a capsule, was it steam sterilized or gamma sterilized? So there's some information on the sterilization process. And then what are the acceptance limits of the analytical methods that assure, again, the requirements for identity, strength, quality, purity of that drug product. They describe those methods. Again, if you're using a third party, or even if this is something that you do yourself, certificates of analysis are highly regarded by the FDA. Next slide then. So information is the type of stability of the drug product. You include data on stability and test methods for the packaged product. So you do this for the drug substance itself, but also as it's put into whatever system, the container system that is used. Make sure that it is stable in that container system. And it's important to show that stability, the drug product was stable for those toxicology studies that you did and for the proposed human clinical studies for the duration of that first human study that you're proposing. So here's something that's kind of unrelated to your actual drug, but if you're using a placebo, you need to have a description of its composition, where it comes from, who's making it, and so on. And then copies of all labels that are going to be provided to the investigator during the clinical trials. And something that the FDA requires, in addition to any information that you want to put on the label, you have to have that statement there, caution, new drug, limited by the federal law to investigational use. So that has to be on the label. And then there is a portion of this, the claim for exclusion for environmental assessment. So this comes out of the congressional law that requires all government entities, all government groups to provide the environmental impact statement for any action that they did. When they issue you an IND, that's an action. So this is actually pretty easy. There's verbiage on the FDA website that you can put into your CMC statement. But they just want to make sure that you're not letting, the sewer system for the drug is going to cause some sort of problem. So it's required, but it's pretty easy to do. Okay, so that's a general list of the elements that are required for a CMC section. So now we're going to go back and talk about some strategy and what needs to be done in detail for some of these different processes that you go through in the development of the drug. So again, this slide's in here to reiterate the idea that you really start putting CMC information together early in the drug development process. So there's a variety of things that you do in that preclinical phase. That's phase before you get the clinical development. So formulation, for instance, you're trying to figure out how you want to formulate that drug so that it's going to be stable, and it's not going to cause an issue if you're injecting an IV into somebody's arm and so forth. So all these attributes that you can find out in working through these preclinical stages become relevant to CMC. Next slide. Then. So the amount of information that you need to put in that CMC is going to depend on a variety of things, and they're listed here. So what's the phase of the investigation? So if you're going into a first-in-human studies, a phase 1 clinical trial, as we said, you don't need all the validation data. How novel is the drug? Is there something is this a second generation? You've got a lot of information on the first generation. So maybe some of that early phase, that first generation drug information can be used for this second generation. Are there any previous studies? Are you using this same drug for different indications? So it's a new clinical trial, but you've got a lot of the toxicology and the other information already developed for that drug because of those previous studies. What's the dosage form, the route of administration that can change what you need to put in? What's the duration of your first clinical study? And what's the patient population? Is it a relatively modest population, low number of patients? And are there any known or suspected risks? If it's a pretty safe drug, where there are no flag interrupter signals that came up in the preclinical work, maybe you don't need to put in a lot of extra information in the CMC section. So it depends on a variety of things that are listed there. So these are that was kind of a transition slide. So these are the various elements of the development process that we're going to go through in a little more detail here. So in your development phase, when you're working with your lead compound and trying to think about how you're going to formulate this, you go through this pre-formulation phase, and what you're actually going to manufacture in the long run is going to depend on the properties of that drug substance. So information you use is based on your characterization of the drug substance, even in those early stages. So there's a focus on predicting the formulation choices of the drug based on things like its drug form. So is it going to be a solvent? Are you lyophilizing it? So if you're lyophilizing it, you need things to stabilize it as it goes through that initial freeze, as it thaws out, and how stable do you want it to be in a solution while it's sitting, for instance, in a type of syringe? So what's the dosage form? Excipients, what are you going to need to put in there to maintain the stability of the drug, to maintain the pH of the drug before it's dosed to the patient? You're considering and you're evaluating a number of these things as you're going through your pre-formulation. Okay, next slide. I forgot just about those. So the next couple of slides talk specifically about small molecules versus biopharmaceuticals in terms of the physical characteristics and how you're going to formulate these things. So if it's a small molecule, what's the particle size? Does it dissolve very easily? What's its purity? And you can define that by melting point or other characteristics. What's the solubility in various solvents? What's the pKa dissociation constants if it's an amine group or carboxyl groups? What's its membrane permeability, which can determine how readily it gets inside a cell if that's where the target is. And what's its lipophilicity? So is it going to be something that's going to be absorbed very readily from the gut, for instance? So those are some characteristics that you want to address. Next slide, then. For proteins, there's a variety of characteristics that are normally included in the description of those proteins that are part of your pre-formulation thought process and what goes into defining the nature of your protein in your CMC. So what's its isoelectric point? You want to avoid that because that's the point of where it's least soluble. What's its thermal stability? Something about its structure, primary, secondary, tertiary structure, any post-translational modifications that are important. Glycosylation is something that became very important in the 90s and the 2000s as people realized that it could affect the properties of biopharmaceuticals, especially monoclonal antibodies. And glycosylation is one of those things that is very dependent on how you're making your protein. So glycosylation analysis became a big area for not only investigation, but incorporation into assays that were developed in biopharmaceuticals. What are the impurities and the degradation products that are in your protein? And that's also important for small molecules too, by the way. But for Baxter, we always had to worry about what was leaching off of affinity columns and what was coming through in the processing from the CHO cell, for instance. That was the cell line. So the FDA requires some base level. You can't go above some nanogram per mil level of CHO cell host protein. And then what's the solubility? Clearly, if you're getting a protein that you have dilution, you don't want it precipitating out on some of the shelf. So what's the solubility, and how do you preserve that solubility on some of the kinds of excipients you put in? Next slide there. How are you going to deliver it? And so you can see this list of various ways of delivering the drug, and there's a whole range of them. Each of them require their own unique formulations. And so what are those things that you're going to put in there that you're going to define in your CMC that are going to allow you to deliver it in that format? So I don't think I need to say any more about that, but you can certainly look at that list and see that it's quite a few ways of doing it. Okay, next slide. And so early on, you're developing a whole array of processes and conditions and things that you want to meet that include not only the characterization of the protein itself, but also aspects of how this is going to be used in the marketplace. It can affect the way you formulate. So compliance with regulatory, or not only the stability, but you want to minimize medication errors. You want to make sure that this thing is manufacturable, or it can be manufactured at a lower cost. So these things all have impacts on the way that things are formulated. If you want to hit the button, we'll get the bullet points in there.
Oh,
So,
hang on.
can
Flash.
you go back?
There you go.
Yeah, there you go. So they're developed to evaluate, a whole array of things, including, the stability of the actual product, but what are the clinical needs? How are you going to deliver this? What's the cost of these formulations? You're making something that you want to be as gentle and easy for the patient as possible. So you're taking those sort of things into account. So you're going to develop multiple formulations with, that can be addressed across a variety of these properties, and pick the one that works best for you. And all of this information is going to end up, or the final formulation is going to end up in your CMC. Next slide.
There we go. Okay.
And then this is something that generally is discussed very in a very detailed manner in your preclinical side of your IND. But how a drug is absorbed determines its dose, and those considerations can be part of the CMC. So there are aspects of how it's delivered, how it's formulated, that affect the absorption from the gut, and sometimes even how it can be cleared from the body, whether it's a particular type of salt and so on. So these are other aspects that you might consider in pre-formulation. And so these are the processes that you're going to go through in your development process. And Ed and I are going to talk about some of these things in a little more detail. Some of this will be redundant, but I think it's good to cover in a thorough manner so you really understand how these things fit into your CMC. So analytical methods. Next slide. So the federal law requires you to define the identity, purity, and quality, right, of your product. Not only the drug itself, but the drug product. So strength, you're measuring the concentration of potency in that substance. Identity, you're using things which uniquely identify it as the drug, so active PLC and so on. You quantitatively measure the drug purity and show that if it does have impurities, you've already shown by preclinical studies those impurities don't affect the efficacy of the drug or have any toxicity. So it's okay to have impurities in there. They just need to be consistent, and they need to be addressed from a toxicology perspective. And then the quality measures that are put in place to ensure that you've got a safe and efficacious drug. Next slide. So product release is one of those aspects that where you're addressing, again, a number of these parameters that the FDA requires you to talk about in your IND. So potency, what's the biological activity of the drug, what's its concentration, how have you identified it, you've shown that it's pure. The impurities here, you can get degradation from small molecules and biopharmaceuticals. So, what are the degradation processes that occurs when it gets at stability? It's not even going to do. Solution properties, sterility is one of those things we haven't talked much about, but you have, obviously, when you're making your final product and you're releasing it, you have to demonstrate that your sterility process works. Whether you're using sterile filter or gamma radiation, maybe you're doing an endotoxin LAL test, you're looking for bioburden by doing bacterial cultures, or there's some other, they used to use a lot of rabbit pyrogen studies at Baxter. The FDA has asked people to get away from a lot of this, so I don't think much of that is done anymore, but what are there are other tests that we were developing to substitute for that, but you've got to develop really to show that your product is in fact sterile. And the details of those are part of your sample. Next slide. So again, the attributes on the left side and some examples of specific analytical methods. You guys probably are pretty well versed with most of these, I would think. So the rapid power test is still listed, but I think most companies are trying to get away from that test using other tests such as the monocyte activation test and so on. Next slide.
I'm trying. There we go.
And then potency assays for biopharmaceutical are listed here. A lot of you guys probably know most of these, but I'll go through them pretty quick. So if you're dealing with an antibody, you may have a binding assay, or a receptor agonist, you may have a binding assay. Enzyme assays, of course, are pretty straightforward. Bioassays, if you're got a whole cell, animal, whole organ, or tissue culture where you can look for some sort of an output. And of course, immunoassays, where you're using an antibody in an ELISA format to assay the protein. Next slide. So in the manufacturing process, there's a variety of steps that you go through. In that, there's an analytical test that need to be done to assure that you're getting the protein that you want or the small molecule that you want. Well, these are really required pharmaceuticals. So the protein that you want during these various stages of the production. You can see those listed over on the website. Most of these are pretty standard, but they are tests that you're going to have to describe and show that they work. You're going to have to validate them at some point, and all that information you're seeing.